Okay. Hi everyone. Good morning, and thank you for being here. It's for me a pleasure to introduce Professor Peter Tieleman. He studied physical chemistry at the University of Groningen in the Netherlands, where he obtained his PhD under the supervision of Professor Herman Berendsen, who is one of the pioneers of the biomolecular simulations. After a year as a EMBO fellow at the University of Oxford in the Mark Sansom research group, Tieleman joined the University of Calgary in 2000, and he is currently there a professor and a researcher in molecular simulations in the Department of Biological Science. He is working in the area of biomolecular simulations and computational biology, with a particular interest in cell membranes, where he is a really recognized and well-cited researcher. So, Professor Tieleman.
Thank you. Thanks for the introduction. Thanks for the opportunity to come visit here, even though I understand the timing is not perfect. So far, I've had a very interesting visit. I'm going to talk about our work on cell membrane models. This is Calgary. We're close to mountains too. They're not quite as high, but they're very good at snow and they're much colder. So this is a textbook picture of a cell membrane. Or maybe it's a bit of an old-fashioned textbook picture. You have a lipid bilayer that forms the main structure. Then you have proteins that are part of the membrane, these black blobs here or the red blobs here. And then you have additional structures, cytoskeleton that gives the cell mechanical properties, and it has carbohydrate layers and other recognition elements. So this is biologically a very complicated system. This picture is from textbooks that have been around for quite a while. And I think the actual picture you would draw now is quite a bit more complicated, and I'll show a computational model towards the end of what it might look like if you actually would draw proteins to scale. In particular, the protein density here is quite low compared to what we currently believe is the case. We're interested in calculating and making models of a cell membrane and then use those to answer questions about biological phenomena occurring at the cell membrane. And these are quite important. People always cite in grant proposals and abstracts that about a third of all drugs is, or no, more than half of all drugs is targeted at proteins in cell membranes. About a third of all proteins is in cell membranes. So they perform communication functions between the inside of the cell and the outside. They transmit information, they transmit metabolites, the nutrients, signals of various kinds. So this is a state-of-the-art cell membrane model from about 20 years ago from my PhD. This is a single lipid, so the complexity is greatly reduced to our cartoons from the previous slide. But this is actually already quite nice in many ways. If you think about the structure of a lipid bilayer, this is probably the most accurate structure you will ever see. So experimentally you cannot measure this, you cannot see this structure because the lipids are disordered. You can calculate, we can measure average properties. And two common techniques are X-ray diffraction and neutron diffraction that gives you a distribution of atom types along the normal on the membrane. And deuterium NMR that gives you an average degree of order of all of the CH bonds in a lipid as a function of their location on the lipid. So those are properties that give you structural information, but not very detailed structural information. If you use those properties to validate or to develop computer models that give the same average properties as you can measure experimentally, you can then use computer simulations to get this very detailed image of what lipid molecules actually are doing over time and their average structure. So computer simulations add a lot of detail to experimental data. And they're validated by experimental data as well. I think many of you here are well familiar with computer simulations, but just a brief description of how this works. We make a description of all of the atoms in the system. So this is chemically accurate in the sense that every hydrogen, oxygen, carbon is in the model. We describe in an approximate empirical way how these atoms interact. We know the accurate way. Quantum mechanics describes this to almost arbitrary accuracy, but only for very small numbers of atoms. So we make a simpler description that mimics the properties that you would calculate from quantum mechanics. Everything okay?
Yeah.
Okay. And then you can calculate how things move. So for the physics department, this is embarrassing because this is high school physics. This is basically saying, okay, you have a brick and I go to drop the brick. What's the velocity, the position, and the acceleration at any point in time? So you can predict, or you can calculate the trajectory of the brick, or cannonballs, or again, high school physics. But we do this for tens of thousands. This is about 8,000 atoms. To now, I have an example with 130 million atoms. So this becomes a computational problem that even though the physics is simple, the computer science part behind this gets quite involved. This movie showed dynamics on a 100 picosecond timescale. And that's one other thing that's interesting about these simulations. We're interested in timescales up to milliseconds or seconds, so macroscopic timescales. But we use integration time steps of femtoseconds. So you need thousands of billions or millions of billions of steps to actually get to that macroscopic timescale. Also, this means that the reach of our simulations increases with increasing computer power. So this is state of the art 20 years ago. But now if you go to a few years ago, 5 years ago, we moved from 100 picoseconds to microseconds. What that means in terms of lipids is you can look at lipid mixing. So you can look at mixtures of lipids. And one of the goals of these two simulations was to look at how much time you actually need to do meaningful simulations of a two-component mixture. This is actually kind of depressing if you think about this in some way, depending on your point of view. The movie I showed from 1996 was actually pretty nice for a single lipid component. But now if you're interested in mixtures, it takes microseconds to get statistical sampling of a mixture. And people have tried to improve this with various non-equilibrium techniques without much success. So basically you just need to sample microseconds. The nice thing is 20 years later, for orders of magnitude to computer power, that's actually readily accessible on GPU, single GPUs, and you can do more on large supercomputers. So that's one thing, microseconds for lipid mixtures, and then you can do questions like, if I artificially separate a binary mixture, this is POPG, POPE, which looks like a bacterial membrane, so biologically this is inspired by questions on bacterial membranes, or cholesterol and POPC, which looks like a eukaryotic membrane, red blood cell or any higher organism. You can ask, how long does it take to become converged? Microseconds. And then you can ask, does this actually have a structure other than random? That's not trivial, because you could imagine that these blue lipids or the cholesterols like to cluster compared to being randomly distributed. So we didn't really know the answer. And experiments can't really measure this. They can measure phase diagrams and they can measure average order parameters, average cholesterol orientations, but they can't look at very small clusters of lipids. So if you look at these two pictures, let's look at this one first. I'm going to take a vote. Which one of you thinks this is random? So the question is, so this is the equilibrium distribution, or a snapshot of an equilibrium distribution of this binary mixture. Do you think this has a structure in it, or is this a random distribution of blue and brown lipids? So, anybody want to vote for random? No? You? Brave of you. Anyone else? So everybody else is voting for this as a structure. So this is actually something to worry about in simulations. It's very difficult to tell. So if you look at this slide, we analyzed this in some detail. If you look at local snapshots, oh look, these blue lipids are clustering, or the cholesterols here form a cluster. But actually, if you take two components and you put them on a grid, you actually get clusters randomly. So what you need to do to analyze this is to compare this to what you would expect in terms of cluster sizes from placing things randomly, comparing that to what you get from the simulation. And if you do that, these are cluster size distributions, there is no difference between what we see, and a random distribution. So you win. So this is actually random. And there's a bigger point here. People looking at simulations, these have become very accessible, not just lipid simulations, but protein simulations, tend to look at snapshots, especially if you're not used to thinking about ensembles and averages. They look at snapshots and say, oh, look, this hydrogen bond here, or this cluster here. But that's very often just looking at anecdotes and looking for what you want to see. So your eye is very good at finding patterns in things that don't actually have patterns. Okay, so microseconds on binary mixtures, but that's still a very limited thing if you want to move to the cartoons at the beginning of my slides. Real membranes have 1,000 lipid types, maybe 50 or 60 lipid types that are more than 1% of the membrane. There are many very rare ones that have specialized functions. And real membranes have lots of proteins. But this continues to be very difficult at this level of resolution where you have every single hydrogen, oxygen, carbon. So for the last 12 years or so, we've collaborated with CEO Mark at the University of Groningen, who developed a coarse-grained force field that was meant initially to look at lipids, but now is able to look at almost any chemical system. And in 2007, this force field was called Martini. This is a reference to cocktails, but also to the church of the city of Groningen, where both CEO and I come from originally. So in Martini, you combine 4 atoms on average in a single interaction site. So you lose detail, but now you also make the number of particles much less, and the scope of simulations can become much larger, up to a factor of 1,000 in many cases. So you can look at a similar system, but now for 1,000 times longer. So you go from microseconds to milliseconds, or you go from 20,000 atoms to 20 million atoms with the same kind of computational cost. One of the big tricks in this coarse graining business, and this has become quite popular in recent years, it's not new. Polymer chemists have been doing this forever. Polymer chemists would think you're crazy if you describe every hydrogen and every carbon. So they've used simplified models forever. But in biomolecular simulation, this is relatively new, has been a big thing in the past 10 years or so. But these are all of the amino acids in the Martini representation. And I'm showing this to give you an idea of the level of resolution that you can still achieve. So isoleucine and leucine look the same, but if you look at the numbers in the tables in the software, one is slightly darker blue than the other. But having said that, if you're interested in leucine zipper packing, and you're worried about the difference between isoleucine or leucine and valine, Martini is not a good choice. Because you lose details of packing, and you lose all kinds of other details, but you gain a lot of speed, and for lipids, oriented problems, this is actually very useful. So we've been working on tools. This is a slide from Tjerk Wassenaar, who's been both in my group and in CEO Jan's group. He's currently still in CEO Jan's group. This is Martini. This is atomistic, so every single atom. And this is an even coarser model that membrane biophysics types, especially from the real physics community, like, because you can do mechanical calculations on this, bending, and you can make very large structures. But this is very simplified. You lose all chemical detail. But we've worked on protocols where you can actually go from this to this. And Martini would be an intermediate in this case, or from Martini to atomistic. So the idea is that you can do detailed simulations, and then if you want faster sampling, you could go to Martini. You could use Martini to sample lipids moving around or proteins moving around, and potentially go back to atomistic and ask for more detailed questions. So with Martini, I showed this 2 microsecond binary mixture. This is a paper from the same time, 5 years ago, from Helgi Ingolfsson. This is a model of a plasma membrane of a eukaryotic cell, like a human cell, that has 60-something components. So all the major components of a typical plasma membrane are represented in this. Plasma membranes are asymmetric. The inside of the cell has a different lipid composition than the leaflet on the outside, and that's also captured by this model. And now we can do simulations not of 2 microseconds, but of the order of 100 microseconds. So we're using the faster speed of Martini to go both from 2 microseconds to 100 microseconds, or 80, and we go from a few hundred lipids to thousands of lipids. And now you can ask questions about the structure of a real lipid mixture or a biologically relevant lipid mixture. Does this have non-random elements? Does this have some kind of organizational principle based on lipid-lipid interactions? Well, that's much harder to see, although it's clear that these red ones are clustering. And that's not just your eye saying that. This is actually happening in the simulations. These are ganglioside lipids, so one of the many components in the bilayer. Now you have to decide how to analyze this. So Camilo's group has looked at lipid maps or ways of looking at density profiles. I'm quite interested in hearing a bit more about that and seeing that. Because that becomes the way you analyze these complex mixtures. You don't look at individual lipids, but you calculate properties averaged over many lipids. So this shows, for instance, as a running average over 200 nanoseconds, how certain groups of lipids have densities that are more or less than you expect based on average. So if the lipids were homogeneously distributed all the time, all of these graphs would just be white. There would be no features in them. But they're not. Any individual snapshot has a structure. And you expect that because it's not going to be random in each snapshot. It will actually have local clusters. Over time, you expect those to disappear. But if you get persistent clusters, for instance, these gangliosides actually cluster together and stay together, you would see that in movies like this. So what we find is, just summarizing, the structure depends on averaging time. That makes sense. On a very long time scale, you expect almost all the structure to be on average 0, but you could still have local structure for parts of the simulation. On microsecond timescales, almost every structure disappears in these mixtures. And that's relevant in biology because people are arguing about transient small clusters of lipids that are biologically relevant for platforms for signaling. But if those disappear on a microsecond timescale, that's probably not enough to have a functional role. So that means either these clusters don't exist or they have a different reason for coming into existence that's not captured in these simulations. And that's quite possible because we have no proteins and we have no cytoskeleton. We also have no active elements. So there are things that are not in this simulation that the real cell membranes have. I mentioned gangliosides, PIP lipids also cluster. And then there was a story about cholesterol. Cholesterol actually flip-flops between the two different leaflets. And there's an experimental discussion at the moment, as well as with simulations. If you take a red blood cell or any other eukaryotic cell membrane, what is the concentration of cholesterol in the inner leaflet versus the outer leaflet? The composition is not the same, so the concentration doesn't have to be the same. And there are transporters that move cholesterol around potentially from one leaflet to the other. So at equilibrium, a cell membrane would be dead. It would have the same composition on both sides. But that never happens in real cells. So some experiments show that cholesterol is highly enriched in the outer leaflet. And there was one controversial paper that says 92% of the cholesterol is on the outside, 8% on the inside. There are other experiments that say it's upside down. There's more cholesterol on the inside than on the outside. So this is a question that continues to be debated. You can look at this by atomistic simulations as well. These are a couple of snapshots from cholesterols that on a microsecond timescale actually flip from one side to the other. So I think we can look at this in more detail in the future based on both Martini simulations and atomistic simulations. This is actually from quite a complicated set of atomistic simulations that is actually recent, 5 years beyond my 2 microsecond movie. This is 10, 20, 30 microseconds on a much larger system on a bilayer with multiple components.
I'm sorry, would this be like natural flip-flop or like just randomly or like with light flip paces or in flow paces like proteins that actually move them around?
So in this case, I don't think cholesterol is flipped by flip paces. There are transporters that have cholesterol as a substrate, but they tend to be involved in cholesterol metabolism. They move cholesterol out of a membrane into HDL and LDL particles for biological reasons. Flipases and scramlases tend to move specific phospholipids. So cholesterol, I think, will flip readily on a sub-millisecond timescale between leaflets. So it's almost impossible to have a big gradient between two leaflets. So this 8% division would require some active element that keeps flipping cholesterol from one side to the other. And people have estimated how much energy that would cost. It would be most of the energy a cell generates. Or it would mean that the chemical nature of the two leaflets is so different that the affinity for cholesterol for the outer leaflet is very much higher than for the inner leaflet. And that's not plausible either, all based on simulations. But I would argue, even based on chemical intuition, that seems unlikely. Okay, so now these are all pure lipids. We've been looking at membrane proteins. So if you add membrane proteins to this complex lipid mixture, you can actually look at how lipids redistribute around these proteins. So we have a whole panel of proteins. This was a choice I made 5 years ago. These are all eukaryotic proteins, and they represent different families. So their particular functions are not so relevant in this story, but if I would try this again now, and we are trying this on other cell types, I would try to pick proteins that are closer in real life in the same cell type. For instance, we have models at the moment of cardiac cells. And that's possible now because there have been so many new membrane protein experimental structures. We know most of the structures of cardiac cells. We know most of the structures of red blood cells. So rather than a generic model with proteins that are kind of randomly taken from different cell types, at the moment you can make cells that look pretty realistic in terms of specific proteins that actually are in cardiac cells or neuronal cells or red blood cells. So we're going to stuff these into bilayers with the same composition as the plasmid membrane I showed. And now we're going to ask a couple of questions. So one is from a simulation point of view. If I put these proteins in a lipid mixture, this system will be out of equilibrium because it's very likely that these lipids would like to have a different distribution around the proteins. So how long does it take to look at lipids rearranging around proteins based on the interactions between lipids and the proteins? And this is one way of addressing that. The answer is of the order of 25, 30 microseconds in Martini. So if you want to try this atomistically, for technical reasons, this is probably closer to 100 microseconds. So if you're interested in this, you have to assess whether you have the computer power to actually do this. And then one of the main details you get out of this are graphs like this and graphs like this. So this is a table of enrichment values. So we calculate after the simulation, averaged over the last part, what the concentration is of each protein, each lipid type around the protein, and we compare that to the bulk concentration. So if this would be the same, the protein has no effect on the local lipid concentration. And the enrichment factors all here would be 1. But certain lipids are probably attracted by the protein, and others might be either repelled or outcompeted by the ones that like the protein. So you get numbers like this. 1.8 is almost twice the concentration in bulk for polyunsaturated lipids. 0.8 for another large group of lipids, so they're excluded from the local environment of the protein. And you can do this for all of the proteins, and you get spreadsheets full of data. But there are 2 points that I want to highlight here. So these are enrichment factors for all of the proteins compared to all of groups of lipids characterized by chemical similarity. But you can do this for each type, and you can group them any way you like. This paper was published last year, and there's a spreadsheet. So if you want to play with this, be my guest. But there are 2 points here. So one is that for all proteins, the lipid composition around the protein is different from the bulk lipid concentration. So the proteins change the local lipid concentration compared to bulk. The other thing that was interesting is that if you compare all of the proteins against each other, the lipids around each protein have a statistically different composition compared to all the other proteins. So each protein generates its own unique lipid environment. And you can analyze this in different ways. This is looking at lipid densities around aquaporin. Used to be a local favorite here, might still be, I don't know. A potassium channel, Kv 1.2. And you can actually look at local densities. So red means it's enhanced, blue means it's lower. So around aquaporin, there's an area where you see more polyunsaturated lipids. And then there's areas where you don't see polyunsaturated lipids, but you see fully saturated lipids. If you don't know what these things mean, they're just chemically different lipids. But biologically, this is potentially interesting. Then there are a couple of sites where you actually see cholesterol molecules with a high density. So there are sites where cholesterol seems to bind to aquaporin. You can also look at properties of the membrane, mechanical properties. So if you look at the thickness, aquaporin is one of the more boring cases. It has little effect on the thickness. But around proteins, the membrane thickness can change based on these different lipids. And you can calculate the mechanical properties like the Gaussian and the mean bending modulus. So you can actually look at how the membrane is locally bending around the protein. And I think that's one of the big forces that might shape the distribution of proteins in addition to direct interactions. So mean and Gaussian curvatures, this is an example of 4 of the proteins that we analyzed in more detail. Aquaporin has relatively small effect. It looks pretty, but if you look from further away, you just basically see a flat membrane. And if you put aquaporins in a large simulation, the membrane stays mostly flat. This is P-glycoprotein, another large dimeric protein, and this looks more like a dipole. So actually, if you would look at this, you could imagine that this would induce these proteins to line up, because they would bend the membrane in the same way. And that's actually what you see. If you put lots of P-glycoproteins in the membrane starts bending in a very strong way. So now if you would have a competition between these and these, or between all 4 of these, you get potentially unpredictable effects, although you could calculate them from simulations. This is an interesting feature. COX-1 is the only protein out of the 10 that's not transmembrane. It binds to the outside of the membrane. But it actually affects the inner leaflet as well as the outer leaflet. So this is the outer leaflet, but you can actually look at the inner leaflet, and there too the protein bends the membrane, and the lipid composition changes on the inside. So there are many proteins in biology that bind on one side of the membrane, but clearly interactions through the membrane are possible and might affect the way they function biologically and how they might transfer information to the inside. If you're interested in details of proteins, you can try to link that to the properties that I just showed, these density maps or curvature maps. So this is P-glycoprotein. It's a large protein. This is the transmembrane part. And these are ATP binding domains and ATP hydrolyzing domains. And if you're interested in details of P-glycoprotein, and there are thousands of papers that are interested in specific residues in P-glycoprotein and their role in transport, you can correlate the structure with the properties I just showed. I won't go into any detail here, but if you have a pet protein where you think it would be interesting to look at lipid distributions and specific mutations, that's definitely possible. This is P-glycoprotein again. This is now a density map of cholesterol looking down on the membrane around the protein. So cholesterol is enriched around P-glycoprotein. And also, phospholipids are actually inside the cavity in the protein. So you find 3 binding sites for phospholipids that are very well defined. This is not something you can get from the crystal structure very readily, because there are no structures there, no lipid structures. But this is a very dominant feature. And you can actually look at this by simulations. And we have a paper 2 years ago from Estefania, who calculated lipids moving in and out of this protein on a 20 microsecond time scale, and you can basically see pathways of lipids going in and out, and you can calculate rates if you want to for that. These are 2 other examples where you can actually find local densities, some of which are also present in crystal structures or EM structures. So there is a way to validate these simulations to some degree compared to experiments. This is an example of a more detailed and more recent project. This is a potassium channel that has electrophysiological properties you can measure as a function of cholesterol concentration or mutations. So it has conductance and an open time, all the usual things people can measure accurately from electrophysiology. And what these things show are cholesterol densities mapped on the surface of the protein. So the question that we're trying to address here is, how does this cholesterol distribution change based on mutations? And can we try to correlate that to what you could measure by electrophysiology? But this is work in progress, and I don't have a complete story here. But this is just an indication of going from how you can go from this generic protein panel and generic mixtures of lipids to specific questions on, in this case, the effect of cholesterol on the functional properties of an ion channel, and the effect of mutations on the effect on cholesterol on functional properties of ion channels. So I don't know whether this is going to work, and I think very likely you will need atomistic simulations in addition to Martini simulations, and we have atomistic simulations here. But I think this is a direction that will be useful in the future. So I have 2 conclusion slides, and then I have a few more slides about a different topic, just because I think that's a cool story. I think there's still time, right? Yeah. So conclusions from the lipid-protein story. Each protein has a unique interaction fingerprint in the complex mixture. So that was the main point. Every lipid environment around the protein is different from bulk, but also from other proteins. There's a lot of emphasis on the literature on specific lipid-protein interactions. A lot of this is based on structural studies that see 1 or 2 lipids bound, or on mass spec studies that identify in a complex of a protein flying in a mass spec instrument certain lipids. And some of those are clearly functionally relevant. So one example actually are the ion channels I just showed. The general family of amide rectifiers is activated by PIP lipids, and without them they don't really function. So that's a clear example of where you need lipids functionally. But these enrichments and depletion factors, even if you remove 20% of a specific large class of lipids around the protein, that's actually a very large effect. The binding affinity for these lipids is very small. You wouldn't measure it in a mass spec experiment. But modifying the average environment around the protein by the sum of many of these small interactions, I think is a very strong driving force for membrane structure. And one way of looking at that is taking these Gaussian curvature maps or mean curvature maps and see how they compete if you have a mixture of proteins. From a simulation point of view, some things we learned are kind of annoying because they're expensive, but on the other hand, 5 years from now this will be easy. Tens of microseconds are basically a minimum time you need to be able to simulate to look at lipid distributions. And if you have small percentage components like diacylglycerols with a 0.8% prevalence in the membrane, sampling becomes much harder. So you need to be worried about that. And then from the curvature maps and density maps, membrane perturbation by proteins can be quite long ranged. People have argued this before, but you see this in a very striking way in our density maps, where the local lipid densities around the protein still have features nanometers out, going into the bulk. So in principle, you could have proteins talk to each other even when they're separated by 5 or 10 nanometers in some cases, like pig glycoprotein. So some of the things that we're looking at in this area, improvements to Martini, and maybe we can talk about that tomorrow in a little bit more detail. Looking for experimental validation of some of our results on lipid-protein interactions. And that's a weak point in this whole story. There isn't a lot of experimental data or experimental techniques that are able to look at these interactions, these weaker interactions in detail. We can use the same methods to look at specific proteins. So I showed this potassium channel, and I flashed to other proteins. So look, you can do this on this dopamine transporter. And so if you have specific questions about specific proteins, and this would typically be in collaboration with people who spend a lot of time looking at specific proteins, you can do this. You can look at conformational changes. And Martini doesn't actually incorporate conformational changes, but you can model proteins in different states in some cases. So pig glycoprotein, as an example, it has essentially 2 states, one like this and one like this. So you can have, there are structures of both states now. So you can take those structures and look at differences in lipid interactions or whatever question you're interested in. And we have a manuscript that looks at a large number of GPCRs from different families. And we look at whether the lipid environment around these GPCRs is different for each GPCR, or whether you can group them into families. And one of the things we're finding is that if you look at the different families of GPCRs, the lipid environments are more similar around GPCRs in one family compared to others. So that might be interesting. Also, we're looking at a number of simple models again, small bacterial proteins and VOLP and peptides, to test whether we get the same distributions from atomistic simulations and Martini simulations. So for what I showed, these systems are too complicated to get sampling atomistically, but you can make systems that have maybe 4 lipid components with high concentrations and no small components. On smaller systems, like 10 by 10 nanometers, maybe in aquaporin, where you can get atomistic simulations on a time scale of 100 microseconds. So that you can calculate distributions of lipids and compare them to Martini. So I started with showing cartoons from textbooks. This is a cartoon from a simulation. This is made by Jan Martin's group, but it's a snapshot that goes with a review we wrote in an issue of Chemical Reviews on biomembrane structure that appeared in May this year. So if you're interested in membrane simulations, this is a very good place to start this whole issue, not just our paper. We have 2 in it actually, one on large-scale membrane simulations and one on detailed lipid-protein interactions. So this is probably a more realistic model where the protein density is realistic. The proteins actually have the volume they have because these are based on atomistic structures. So every atom, well, almost every atom is in this model. Lipid composition is as complicated as you would expect from lipidomics data on real cells. And the things we're still missing glycosylation, the carbohydrates sticking out on this side. And you wouldn't be able to tell on this scale, but we also have no cytoskeleton elements in this. So those are still things that you could add. One thing about the cytoskeleton is the attachment point spacing is a little bit controversial experimentally, but it might be 200 to 400 nanometers. So if you do simulations, that should scare you, because having a model that is large enough to have multiple attachment points would have hundreds of millions of atoms at the Martini level, or beads. I want to conclude with a couple of slides about a recent story. It's not really my project. It was dreamt up by Klaus Schulten decades ago, and a lot of people worked on this over several decades. Most notably, recently Alec Aksymiev, Ayman Takharskyt, and Neil Hunter, in the group of Klaus Schulten, of the late Klaus Schulten. So this is looking at the Chromatophore. This is an interesting biophysical system because it does something relatively simple, and it's basically one of the first steps towards life. This thing is alive. This comes from primitive bacteria. But what this does, this is an organelle, basically a spherical thing that sits inside these bacteria that captures light, and from light it makes ATP. And once you have ATP, you can drive all other biological processes. So this is a photosynthetic bacteria that lives in seawater. But this is a cartoon of the process. But basically, you start with light capturing in chlorophylls. This causes chemical reactions where you transfer energy to a quinone pool. And from that, you generate a proton gradient that helps the ATP synthase to make ATP. So it's a relatively simple process. And we know all of the components. So the questions that we're asking, how does energy flow from light harvesting to ATP at the level of basically starting with electrons and photons to ATP synthesis? And if you can model that, you can ask questions about how optimized is the overall chromatophore. And from a technical point of view, this is a serious multiscale problem. You're going from electron capture at a time scale of femtoseconds to ATP synthesis efficiency, which is a macroscopic average property, or even ATP synthesis is a slow process compared to everything else. So you're talking about 15 orders of magnitude in time scale. It's also, if you do simulations, a macho system, and this is the kind of thing that Klaus Schulten would do, and no other person would try probably. This is more than 130 million atoms in atomistic simulation. So this was run on 2 of the largest supercomputers in the US for a very long time. Like very long time meaning long wall clock time. It's run for months on Blue Waters and on Titan. The actual simulation time is 500 nanoseconds, which isn't that long by current literature standards, but it is for a system of 130 million atoms. They even had to modify the software they were using because the file formats were not set up to have that many lines for the atoms. Writing coordinates and reading them takes forever. And visualizing this is a heroic effort that VMD can do. So these are actually not cartoons, these are snapshots from the simulation. And they show the organization of the protein complexes, and they show lipids, and they show quinones and quinols. I'm just going to summarize the techniques we used. The reason why there were so many authors, everybody had their own pet technique, and that many of them had one individual component in this ladder from femtoseconds to ATP efficiency. So this is the most expensive part, an MD simulation of a 136 million atom chromatophore model. Then based on these snapshots from the simulation, there are QMMM calculations that actually looked at light capture on the chromatophores, on the bacteria, with the chlorophylls. Martini simulations were used to look at local bending effects on the vesicle and try to match that to AFM data and electron tomography data and to atomistic simulations where it wasn't so clear they were converged. There's electrostatic calculations. Based on the atomistic structures, you can calculate electrostatic potential. And in the potential, you can do Brownian dynamic simulations using ALEX software, where you can have thousands of quinone molecules swim around and look at how they move from one protein to the next. And these are the key elements that transfer energy from the light harvesting proteins to the ATPase. Then there's some other stuff that looks at more details, umbrella sampling. And I think this is funny. Then there's experiments that uses TLC. Does anybody know what this is? So it's thin layer chromatography. So if you do high school chemistry, you might do this. So this has the most high-tech calculations possible, and then the experiments use TLC, thin layer chromatography, to look at the lipid composition of the of the purified chromatophores. And then there's spectroscopy looking at how many light harvesting complexes 1 and 2 are there. So this is what you get in the end. After an incredible amount of computer time and effort, you get a graph that shows the efficiency of ATP synthesis as a function of light intensity for different salt concentrations. So these are things you can measure, and you can link them to the conditions these bacteria live in. And one of the things that's interesting here, these are actually optimized for low light, because they live in seawater, so they don't get the full amount of sunlight you would get on the surface. And you can actually speculate about how this works and why they have this many ATPases and this many light harvesting complexes and this many other proteins based on what they were optimized by evolution to do in the environment they live in. So I think this is a very cool story. And I just want to highlight Abhi Singaroy, who was the main author on this and who drew all of these things together, did much of the work. He's currently a professor in Arizona. And with that, I'll finish with acknowledging people in my group, in particular Valentina Corrado and Eduardo Mendes Uniandes, who did much of the work on the lipid-protein interactions. I actually involved most of my group, and most of these people actually are no longer in my group, but they were involved in parts of these lipid-protein interaction stories. The ion channels, GERC is a collaboration with Sergei Noskov and Hank Duff and other collaborators, electrophysiologists. And almost everything I talked about has elements of collaboration with Theo Jan Marink from the University of Groningen, Tjerk Wassenaar and Helgi Ingolfsen, who among other things made this plasma membrane model with 60 lipids, but also was involved in the lipid-protein interaction story. Thank you.
Thank you, Professor. And if everyone has any questions, so we have something.
Over there?
Yes. the density of lipids around a protein? I mean, what kind of information can give you about the membrane?
Okay, so I think there are a number of different reasons. There are cases where proteins need specific lipids bound to the protein or close to the protein to be functional. And our approach basically will give, I highlighted weaker interactions, but it will also identify strong interactions. So you can basically put a protein in a membrane and it will tell you these lipids like to be close to the protein. And that might be, you have to link that to other functional data to say, okay, this is what a GPCR needs, or this is what this transporter needs. So that's one thing. The other thing, I didn't highlight it very clearly, but often I phrase my whole research program in trying to understand the properties that give rise to lateral structure in membranes. So we know that membranes are not homogeneous. We are debating exactly what the nature is of local structure in the membrane. So I started with this pure lipids, only single component, that's homogeneous. Then I went to 2 lipids and said, okay, is this random or is there structure here? Then I went to 60 lipids and I said, is this random or is there structure here? So if you now add proteins, protein-protein interactions and protein-lipid interactions are additional factors that might give rise to local structure. And actually, I think they do. I have no data to show that here, but we're looking at even larger scale simulations, well, not larger than the Chromatophore, Martini simulations, with many different proteins, like 200 instead of 4 in a simulation. And you get all kinds of bending effects and local interactions between proteins. So I think lipid-protein interactions and the way proteins modify the mechanical properties of the membrane near themselves actually are an important driving force in inducing structure in membranes. So the lower levels, a single lipid, 2 lipids, 60 lipids don't do very much. But once you add proteins in the mix, you can reshape membranes in pretty powerful ways. And then there would be a next level, the cytoskeleton, which actually I think is the most important component, but that's not in the simulations yet. Does that answer your question?
Yes, thanks.
I have maybe 2 basic small questions. The first one is when you mentioned that you needed like 25 microseconds of sampling and in Martini, and then that would mean like 100 in atomistic. Is that because of the improvement in diffusion coefficient?
Yes, essentially.
So you measure the diffusion and it's four times higher? Because like sometimes it's a very number that can vary quite a bit.
Yeah, so that's a technical point that's important, but there's no easy answer. So initially we said, based on water diffusion in Martini compared to atomistic, and on lipid diffusion in bilayers, 5 microseconds in Martini would give 4 times as much sampling. The diffusion rates are 4 times higher than atomistic. But the problem is that factor is not constant, which is what you're referring to, I think. So actually there are pathetic cases where you have, I think it was alkanes in alkanes. The Martini diffusion is actually slower than atomistic. As far as I know, the only place this is explained or measured is in Dure de Jong's PhD thesis, which you can download from the University of Groningen. It's available. But he has a number of different tests, and these factors are not constant. But for processes limited by lipid diffusion, this factor of 4 is kind of a rule of thumb. One problem with coarse-graining is that different interactions are coarse-grained differently. So not having hydrogen bonds is an obvious thing, and that might change lifetimes. But counterintuitively, these alkanes actually have a larger effect. And you can look at that in more detail. With Martini 3, there are 3 different bead sizes for every type. And you could potentially play with this in more detail, and you can try this for all of the organic universe if you wanted to. So it's a rule of thumb, but it is based, it's relatively accurate for lipid diffusion. If you want to do other things, you need to be more careful. And you can measure all these things, you can test them. But there's no single number.
Yeah, and the other thing was, when you had these maps, like density maps that were showing it's like fingerprint, like that biochemical, I guess, lipid-protein interaction. I think there were a few where well, the proteins, I believe, are very symmetrical. I think it was like a proportion or so.
Yeah.
And but the lipid interaction was not very symmetrical. Is that like a sampling thing? Is that I think or maybe
This one, right?
yeah, for example that.
Yeah, so there are a couple of different things here. So one is we averaged these things over 5 microseconds. If you average over longer, these features become clearer, but we didn't have that much data. We've also symmetrically averaged these graphs, and then we calculated error maps. It becomes difficult to interpret, but you can imagine, I'm not attached to anything, no. You can imagine if you average symmetrically over these 4 subunits, you can basically get a standard deviation for each point, and you can plot standard deviation maps. So you can analyze this in more detail. So I'm pretty confident of the things that I'm showing here are real features. But the degree of error is something that you can argue with, or you can decide what you need. One way of getting this better is simply to take 10 microseconds or 15 microseconds of simulation. And these features will persist, but all of these features will be washed out. So you can tell already that everything is not perfect because this is going to be white on a very long time scale. Because there's no reason why these two would be slightly different. Also, the one problem with color ranges is it's difficult to interpret them to some degree. So these things are all close to 0. So whether it's +2 or -2, plus 0.2 or minus 0.2, to your eye that's very clear. But numerically, it has very little meaning. Just because your eye is very good at recognizing blue versus red. So
but that is something that you can do then, like using the symmetry to like improve or something. Is that something that that is not like breaking the rules?
No. You just have to acknowledge that you did. So actually, these are not symmetrized either. But there is a figure in the paper that is symmetrized, where we rotated the protein. And it works for aquaporin and KV1.2 and a few others. But pig glycoprotein is dimeric, but the 2 subunits are not the same. So it looks symmetrical from at the picture, but chemically they're not the same. So, yeah.
Do you think that understanding these complex interactions in membranes may help clinicians in their understanding of diseases and maybe improve the treatment of them?
I don't know. I would say if it was a grant proposal, yeah, of course. But clinicians are fairly pragmatic, and most of the drugs that they use are decades old. So there's a whole history of computer-aided drug design, and computational chemistry, and combinatorial chemistry, all of which, if you're skeptical, and some people might call me skeptical, haven't really gained very much. So it remains trial and error and large-scale screening that gives most of the drugs. So yeah, we're saying, not just us, the whole field looking at membrane proteins, but looking at GPCRs, and they're very important because they're a target of 30% of all drugs. So 50% of all membrane proteins, 30% GPCRs alone. But there are now dozens of structures of GPCRs since Kobilka got a Nobel Prize for this a few years ago. And I don't think it has made any difference for drug design. Because people continue to modify existing small molecules and do screening on libraries rather than using very detailed knowledge. I still think there's a promise there. And there's a computational, there's a force field in software called AMBER, which you would know. But it was actually marketed in recent years as a drug design workstation. So you can buy an AMBER workstation, a computer with the software and a workflow on it to do small molecule docking and small molecule mutations. And in the past, my understanding is that drug companies were annoyed by the time scale of computational chemistry. If something takes weeks, that's too long. But you can do TI, you can calculate relative binding affinities between 2 drug candidates on these AMBER workstations in a day with almost no knowledge. And if you use Chromax or VMD or NAMD, you can do it in a day too if you know what you're doing. So that might change things. It might make it more interesting to use computation. But I don't know for sure. And the history of this is mixed. So it goes up and down in popularity. I used to work on a project funded by Unilever, which is one of the biggest consumer soap and things like that. I think Dove is a Unilever brand. So Unilever and Johnson & Johnson are the two big consumer not pharmaceuticals, the stuff you buy in a drugstore. I used to work for a project for them, and they had a big computational modeling department, and they were looking at improving enzymes for detergents. And then 6 months after I graduated, they killed the whole department, and they started doing something completely different. And then 5 years later, they rebuilt the department. Who knows? Oh, yeah.
A question about the lipids, the clustering of the some types of lipids. Is this effect also altered by changes in temperature?
Yes.
And how these curves that you have shown there are changing by this effect?
That's it. So we have not measured that, but we have published a paper using Martini at the end of 2017 on ternary mixtures of lipids, where you can go from completely mixed at high temperatures, random, to completely phase separated at low temperatures. And there are intermediate regimes where you can look at domain fluctuations. But at most of those regimes, cluster size is not a very good indicator of interactions. So we looked at the fraction of phase A versus phase B as a function of temperature. And only at high temperatures are these cluster sizes useful. At very low temperatures, they separate completely into two phases. So you would have one cluster of each. So we have tried that. And the cholesterol flip-flop simulation I briefly showed is based on atomistic simulations on the same system. So we have a manuscript at the moment that's being reviewed that uses atomistic simulations of ternary mixtures as a function of temperature. And you can do the same thing. You can go from completely phase separated to completely mixed at high temperatures. And then the intermediate regimes are most interesting, because they see very rich behavior of domain fluctuations and lipids exchanging between one phase and the other. And you could potentially, or you should be able to compare that to phase diagrams and calculate compositions of one lipid in phase A versus phase B and things like that.
When someone wants to look the lipid aggregation, what is the parameter that someone has to measure? For example, the radius or the length of the aggregate.
Yeah, so that's actually that's not an obvious question. We use a combination of parameters to identify a local phase. So it's based on order, the chain order. It's based on the neighbors. So if all of the neighbors are the same type, that makes it more likely to be a different phase. If all of them are randomly type A versus B, that means there is no local domain. And then there are some other parameters that you can tweak to try to identify this. There is no perfect solution for this that I know.
There is a question in the chat, and can you comment on how crowding in biological membranes are changing the lipid-protein interaction?
Yes, well, that's a direction we would like to move to. So I showed this. Sorry. So this is actually a real simulation where there are a few hundred proteins with a density that we think is what you expect experimentally. So protein-protein interactions are very common, and the distance between proteins is less than the range of the effect of the protein on the lipids. So I think in the end, for systems like this, for biological membranes, the lipid properties from pure lipid mixtures are perhaps not all that relevant. But I don't think we really know at the moment. There's a whole area, so that's one other thing, the simulations I just talked about with these temperature-dependent domains, that could be a temperature range that's biologically relevant. But all of the experiments tend to use lipids that are not actually occurring in biology. So they have features that look like biological lipids, but they're convenient synthetic lipids that people use. So how all of these lipid biophysical studies translate to relevant properties for understanding biological membranes, I don't think is a solved question.
I had a bit of a technical question. In the Martini 2, if I'm right, like this year there was a paper where they were showing that it overestimates the hydrophobicity and is not like systematic in the way that it is overestimated. So how it affects the results that you observe on how the lipids interact with the proteins, for example, like, and if then will be necessary to run also atomistic simulations and not just sticking to coarse-grained simulations in those kind of observations.
So I think that's a very complicated question. So I mentioned we're looking at model systems where we can do the same simulations, exactly the same, atomistic and coarse-grained. But there are several different problems here. So the first is, okay, I'm going to make this the first, but atomistic simulations on mixtures are not guaranteed to be reliable at all. So you can do, if you can sample them, which is tricky, but you can potentially run this on Anton or spend the 6 months to get 30 microseconds. We don't know whether they actually have the right mixing behavior. And based on protein simulations, it's actually very doubtful that they have the right mixing behavior exactly. And experimental data on this is not that easy to get. So atomistic simulations are not a perfect way of valid, are not perfect, they're not actually a very reliable way of validating Martini. Experimental data would be. So how problems in Martini affect my distributions, it doesn't take very much to be off by a factor of 2, because that's only KT of the order of KT in accuracy. The point I was making mostly on the generic plasma membrane was that all of the proteins give different environments. And I think that's a much more reliable conclusion than saying this lipid has a 0.8% chance of being here. So that's one thing that helps. In a way, it's cancellation of errors. Martini has been criticized for being too sticky, and maybe that's what you're talking about, or too hydrophobic. So there are different things. Too hydrophobic, the individual beads in Martini have a very accurate solubility in different solvents. That's the basis for parameterization. So the beads themselves shouldn't be too hydrophobic or too hydrophilic, and they should be more accurate than atomistic small molecules in many cases. If you combine this into large molecules, that combination is not guaranteed to be as accurate. There have been several papers that said protein-protein interactions are too sticky. And we see that in these simulations as well. So one way of dealing with that, we have simulations of this system where compared to the published Martini parameters, we scale down the protein-protein interaction parameters by 25%. So now they're less sticky. In discussions about Martini 3, and we had a workshop on this a few weeks ago, I think we know why Martini is too sticky. And actually atomistic simulations have the same problem, because protein-protein interactions are not something that have been tested traditionally because they're difficult to sample. So unfolded proteins and protein-protein interactions both are weaknesses of atomistic force fields. But in Martini 3, I think we can get a better average bead density, and that allows you to get a more accurate stickiness. So the overly sticky interactions now, I think, can be fixed either ad hoc, but in Martini 3 they can be addressed systematically by the way you map the new bead types to proteins. And then the sum of these interactions actually doesn't make it too sticky. That's the plan, at least. So, not an easy question.
I guess there is one last question from the live streaming. And yes, can you expand on the lipid rafts and the need of proteins and or cytoskeletons for their formation?
Sure. So if you look at the literature on lipid rafts, historically these were detergent-resistant fractions, which people now think are an artifact. They've become smaller and smaller, and as microscopes became stronger, super resolution microscope can now look at 40 nanometers, they became transient as well, because there was no clear evidence of lipid rafts under microscopy conditions. So the latest definition was talking about small domains that are transient as well. Which is kind of tricky, because this has made the textbooks, but the evidence for this is very limited. There is functional evidence that these proteins are found together, and they must somehow have a mechanism of coming together. So what I think is the most compelling evidence is work from Mary Kraft, which also is not without criticism by others. What she did was she had cells where she was able to image using an expensive instrument at Lawrence Livermore National Lab, the distribution of cholesterol and sphingomyelin. So one of the most common features of rafts is supposed to be the enrichment in sphingomyelin and cholesterol. And what she showed by her experiments was that sphingomyelin is strongly localized. So you see islands of sphingomyelin, but cholesterol is not. Now there are drugs that you can give to cells that destroy the cytoskeleton. So if you give these drugs to her cells and then image again, the sphingomyelin is no longer localized, it's everywhere. So I think these sphingomyelin cholesterol clusters are in a way not real. They're actually enforced by the cytoskeleton, and the attachment points of the cytoskeleton give a unique local environment that could easily attract a specific lipid like sphingomyelin. And then that would attract cholesterol, but there is so much cholesterol, there's cholesterol everywhere, not just in the sphingomyelin domains. So I think if you have rafts, they're either really transient, like maybe sub-millisecond scale, and small, tens of nanometers, but I think there is a much more powerful organizing force that definitely organizes sphingomyelin, provided by the cytoskeleton attachment points in cells. It's not something that we can address very readily, but I think that's my interpretation of the state of that question at the moment.
Some other questions? Or? So yes, thanks to Professor Tieleman. Professor Tieleman will be here if somebody wants to talk with him later, so you can just come and ask him. So thank you.
There are refreshments outside.
So, hay refrigerio afuera para los que quieran. Gracias por estar acá.